All right, welcome to Chapter 3, the chemical building blocks of life. We're going to specifically focus on carbon, and this is going to go back to a lot of what we talked about in Chapter 2 with the valence electrons. And so what we got to think about is that carbon has 4 valence electrons. It's got number 6 on the periodic table. 2 are in level K. That means there's 4 valence electrons in level L. And so that means it has 4 spots. There, the picture you see has the carbon with 4 little hands, indicating that it can do 4 covalent bonds. And because of that, it can connect up and do lots of different shapes in terms of chains and rings, branches, and balls. A lot of what we see in molecules that are important for life, they all have carbon in them. So the basics of carbon is what we call hydrocarbons. This is where there's a hydrogen located at every single spot where carbon can bond. So what you see down here in the bottom right is the larger balls in the middle are carbon and the smaller ones are hydrogen. So of course a couple of those bonding spots have to be held to be able to have carbon build its structure, binding to other carbons. But anywhere where there's an open spot, that's where hydrogen is going to go. And where we see these are going to be in oils and gases. Not only oils that we use for cooking purposes, but also oils, crude oil, gasolines, things like this are all built off of hydrocarbons. Lots and lots of energy stored in every single one of those covalent bonds. So the other thing we got to know is the functional chemical groups. When we think about chemistry, one of the things that gives molecules their particular functions are these functional groups without sounding redundant. So what we're looking at here is 3 major ones. The hydroxyls, these are going to be the ones that add an OH. So imagine that you have this covalent bond right here, and it's going to bond to a carbon. And when we add that hydroxyl on there, that OH, it gives it certain properties. In this case, it's going to allow it to become more of an alcohol or a carbohydrate. And these are going to then be much more different than the original hydrocarbon that it was when it was just carbon and hydrogens. Another one that we can see here is aminos. Aminos give more of a basic pH to the molecule, and this is going to be the NH2 that we see right here. It also is the beginnings of an amino acid, and that's where the amino acids get their name from, the building blocks for proteins, as we'll see as we go forward. Carboxyls, these are going to give things a more acidic property, so the one right above it here, acetic acid, they've added on the C double bonded to an oxygen, so 2 covalent bonds to that oxygen, and then it has a hydroxyl off to the side, but that whole functional group is what we call a carboxyl, and again, it makes things more acidic, so it's going to be on the other side of amino acid. Amino acid gets its name from the amino group and the carboxyl group. So isomers is in reference that molecules with the same molecular empirical formula but have different structures. And so the idea here is that we're going to see some molecules that have the same exact empirical formula. For instance, glucose has the same formula as fructose, has the same formula as as galactose and cellulose and a couple others. And that's going to be that C6H12O6 that you see right there, referring to having 6 carbons, 12 hydrogens, and 6 oxygens. However, these 2 structures that you see there in that image are what we call stereoisomers, where the backbone is the same, but they differ in the groups that are attached, where they're actually attached to. And so with glucose, it's actually a hexamer, where it's got 6 different points of the ring structure. And fructose is a pentamer, where it has 5 points. But not only that, but the structures that are attached to the different carbons are also slightly different too. However, if you were to actually count up all the carbons, oxygens, and hydrogens you'd see there, they would equal the C6H12O6, thus being an isomer. Another one that we can see is chiral molecules, chiral isomers. These are going to be ones that are mirror images. So imagine, as you'll see here in the next picture, your hands, your thumbs, your hands should be basically mirror images of each other. However, the thumbs are always going to stick out in opposite directions. You're never going to actually get them to sit perfectly on top of each other. And so a lot of molecules in life are in the same vein. We use what's called the D and the L. These are chemistry terms that refer to which way the light bends when it hits a particular crystal of one of these sugars or amino acids. And if it bends to the right, it's called a D molecule. If it bends to the left, it's an L molecule. But the key here is that because so many molecules look the same, the enzymes that are important for doing chemical reactions on those molecules have to be incredibly specific. This means that just a slight difference in a molecule, the enzyme should not be able to recognize it. And that's going to be incredibly important in what we consider to be the lock and key system of enzymes, where the reactant that it's looking for, the chemical molecule that it's looking for, must fit inside the enzyme just like a lock and key, where the reactant is the key, the enzyme is the lock, and if they don't match, then it's not going to open, or in this case, the enzyme is not going to be able to do the chemical reaction that it needs to do on it. So this brings us to some of the same information that you talk about in labs. Most everybody that's in laboratory should have done this lab already. This is the macrobiologically important molecules. And so one of the things that we'll talk about in this chapter is the same material, looking at the carbohydrates, nucleic acids, proteins, and lipids. All four of these macromolecules get their names from whatever their monomers are, which is the pieces that put them together. And then the ultimate polymer is the names that I just called out, the carbs, proteins, lipids, and nucleic acids. So how do they actually form? They form through something called the dehydration reaction. This is going to be exactly what it sounds like. If you're dehydrated, you're losing water. And so what we can see here on the left-hand side, those little white balls are going to be carbons. And what we see is you got a 2-carbon molecule and a 1-carbon molecule. In between them, you have a hydroxyl and a hydrogen, which basically is H2O. H2O gets removed, and now we form a covalent bond between that 2-carbon molecule and the 1-carbon molecule. The same difference is done in reverse to break the molecules up, and this is what we call a hydrolysis reaction. Hydro meaning water, lysis means to cut. And so here water is being added to the system and breaking that covalent bond. The one thing that's missing here is that we should also have an arrow coming out showing that energy is being released from that covalent bond. And in this case, that is going to be the energy that's going to be used by the cell to power the different reactions that need to be done in the body. So the first of the 4 biomolecules we're going to talk about is carbohydrates. Carbohydrates are loosely defined as molecules that have a 1-to-1 ratio of carbon, hydrogen, and oxygen. There's another empirical formula there that you can look at, but the way I like to think about it is for every 1 carbon you have double the amount of hydrogens, and you have the same amount of oxygens as you do carbons. If it fits that paradigm, it is a carbohydrate. So you think about something like glucose that we just looked at. C6H12O6 has double the amount of hydrogens, 12, to every carbon that's there, 6. And then oxygens also equal the same as carbon with 6. So this would fit the ratio of 1-to-1. Carbohydrates have 2 major roles. One is going to be structural and the other one is going to be energy storage. Our body uses carbohydrates mainly as energy storage, but plants also use it as a structural support, as we'll see as we go forward. So the monomer for a carbohydrate is what we call a monosaccharide. It comes from the term one sugar, mono meaning one, saccharide is Latin for sugar. Most of them are going to be 6-carbon sugars that play important roles in biology. However, they can be as small as 3. We'll talk about one called pyruvate when we get up to chapter 6. So, as we talked about, glucose has this formula. Fructose is a structural isomer, and galactose is a stereoisomer. Cellulose, other ones too, all have the same formula. One of the things I want to point out here is that they all end in what? They end in an OSE. Anytime you see an OSE as an ending to a word that you may not be familiar with, your first thought should be that it's probably a sugar. So here's the difference between a structural and a stereoisomer. Structural isomers are going to have one of the carbons is going to have the functional group at a different spot. So in this case, the difference between fructose and glucose, fructose has its double bonded oxygen at carbon number 2, where glucose has its at carbon number 1. So that'd be structural. They're structurally different even though they have the same exact empirical formula. Now, looking at galactose versus glucose, these are what we call stereoisomers because this is going to be more of a mirror image at one spot. So here what you can look at is at carbon 4, the hydroxyl points off to the right in glucose, but points off to the left in galactose. So these are stereoisomers. Either way, what's important for you to recognize here is that those subtle differences are enough for an enzyme not to recognize fructose or galactose if it's actually searching for glucose. And the same thing goes for the other sugars too, is that they are different enough that the enzymes won't fit the lock with that particular key or that particular reactant. So glucose, what is it? It's blood sugar. So this is what people test for when they're testing their blood sugar. It's looking for the glucose levels in the blood. It's a ring structure when it's in its full shape. So we can look at it there on the left-hand side as the linear, where we can look at where the functional groups are attached to and which carbons are attached to. Or we can actually look at it in its full functional shape, which is there in the ring structure. And what we can look at here then also is that the one on the top, alpha glucose, is the traditional blood sugar, where beta glucose is another way to say cellulose. Cellulose is what the cell walls of plants are made up of. Cellulose is what allows vegetable material to, especially like lettuce material, to be a good diet food. Our body doesn't recognize that sugar, so it's filling. It's not bad for us. Our body just doesn't derive any energy out of it, so it can't be absorbed, and therefore, we treat it almost like a diet food. Well, you look here on the actual ring structures, you have the hydroxyl pointing off to the bottom, where in cellulose, it's pointing off to the top. That is the only difference. That is the only difference between those two molecules, one that our body absolutely needs in the form of glucose, and one our body doesn't even recognize in the form of cellulose. Disaccharides are the next step up, so it's just two monosaccharides that have formed together through dehydration synthesis. And so these are going to have a couple common ones that are familiar to everybody. One is going to be standard sucrose. So sucrose is table sugar. This is just sugar that you put in your coffee or put into a recipe, just straight up white sugar. The other one is lactose, which is the milk sugar that's found in not only cow's milk and goat's milk, but also breast milk. Both of these are formed in the same manner. They're going to be formed through dehydration synthesis, as we've seen before. In this case, you look here at the bottom, we have glucose and fructose. The highlighted blue portions there are going to be the water that's going to get removed. So here we have hydroxyl, and we have an H off the other hydroxyl, comes out as water, and forms this oxygen bridge here between the two, and now we have sucrose. So glucose and fructose come together to form sucrose. The other one there, maltose, this is what's found in malts. This is what's found, the sugars that typically are found inside beers and other malt liquors. Polysaccharides is just a name we give, poly just meaning many. This is a name we give for any larger carbohydrates. In this case, these are also going to get the name starches. So when we think about the mono and disaccharides, these are sugars. And when we think of the polysaccharides, we typically think about starches. These are just going to be long chains of monosaccharides. They're going to be put together just like we see there with disaccharide, except for we're just going to have a lot of them all connected together, sometimes upwards of thousands of monosaccharides all connected together through dehydration synthesis. So there's two types. Energy storage. Plants use a starch. Animals use something called glycogen, so we bind all our glucoses together and we store it as glycogen. And as our body needs more glucose and we haven't eaten lately or something, our body will start breaking down glycogen and start ripping off the individual glucose off of there. Plants also use it as structural support. So I already pointed out cellulose. Cellulose is the main component that we'll see as we go forward, that our cell walls of plants are made up of. So we'll be seeing cell walls in the next chapter, in chapter 4. Also, arthropods, in this case, one to always think about is cockroaches or anything that has any sort of insect that has a hard shell. When you step on them or you end up disposing of them in some way, that crunching noise that you actually hear is the chitin. Chitin is the name of the polysaccharide that some insects rely on, as well as some funguses rely on to give them structural support also. So here's just a slide showing you some of those different examples. Here you can just see that over and over again you just get these oxygen bridges that link the different monosaccharides together. The next of the biomolecules we're going to talk about is nucleic acids. Nucleic acids, there's only 2. There's DNA and RNA. DNA stands for deoxyribonucleic acid, and the RNA stands for ribonucleic acid. These are the only 2 nucleic acids that we know of. They are made up of their monomer of nucleotides, so there are only 4 that are found in DNA: guanine, thymine, adenine, and cytosine, or GTAs and Cs. RNA replaces its thymine with uracil. Other than that, it has the other 3. It has guanine, adenine, and cytosine. The structure of a nucleotide has 5 parts to it. It has a sugar, which is where the DNA and RNA gets its name. So deoxyribose, remember OSE means it's a sugar, so deoxyribose, or ribose in RNA. That's going to be the 5-ring carbon, or the 5-carbon ring. There's going to be a phosphate. That phosphate's important for the connection of the nucleotides together to form the chain, so basically making the monomer into a polymer. And then we have the actual nitrogenous base. That's going to be what actually defines a G, T, A, or C. The bases are going to either be purines, which are double ring structures. This is going to be important on how DNA actually holds its shape. And so purines are adenine and guanine. They have a double ring. And pyrimidines are going to be thymine and cytosine. They're going to be a single ring structure. Uracil replaces thymine in RNA. The bond that actually forms between each of the nucleotides using that phosphate, it gets a name called a phosphodiester bond. These phosphodiester bonds are going to be found primarily in nucleotides, in nucleic acids. So here's the structure. You can see the sugar down here at the bottom. You can see it connected to a nitrogenous base. In this case, it'd be a purine because it's a 2-ring structure. We have the phosphate over here, and so this is going to be what it looks like. We name the DNA, so if you go and get your DNA sequenced through 23andMe or Ancestry.com or something, they'll send it back in a GTA and C sequence. And we actually set it up in a way where we're following the carbons from the 5th carbon to the 3rd carbon. And so we name them based off of following clockwise. So when we look here, we have the oxygen and the sugar right here at the top at 12 o'clock. We start going clockwise. The first carbon is number 1. Now, if you're not familiar with this, anytime you have lines coming together in a chemical molecule, the assumption, if there's nothing else there, is that it's a carbon. And so here would be a carbon sitting right there at that number 1 spot. Now that's carbon number 1 because over here at 12 o'clock, it's telling us there's an oxygen there. So we don't assume anything. We know it's an oxygen. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So the ones that we look at the most are going to be carbon 5 connecting to the next, to 3. The next carbon 5 will then connect to this hydroxyl right here, as we'll see. Now, one of the other things I want to point out again is just looking at all these subtle differences. The difference between deoxyribose and ribose comes right there at carbon 2. Deoxyribose, or DNA, has a hydrogen right there. RNA has a hydroxyl. Other than that, they look exactly the same. So yet again, the little changes that make all the difference in the way biological molecules work. So here what we can look at is the phosphodiester bond. So if we remember the 5, the 3' carbon, and how we actually count, we can see here 5, 4, 3. The next one connects to the phosphate, 5, 4, 3. Next one, phosphate, 5, 4, 3. Phosphate, 5, 4, 3. And so we always count our DNA from the 5' to the 3' direction. And that's what you see over here in this arrow, right? You got the 5' heading down to the 3' carbon. You can also see the purines and pyrimidines again. You can see the double ring structures and the single ring structures. Again, this will make more sense as we continue forward with defining how DNA is actually shaped. So what is that DNA is doing? Well, you have to remember the central dogma of biology. DNA is your blueprint. It has all the information you need from the moment that you're conceived to the moment that you die in the form of genes. And these genes are going to be very specific clusters of nucleotides that give off certain pieces of information. Now, that DNA is going to be transcribed into RNA. When we say transcribed, that just means to rewrite. The RNA is just a simple message. It's going to be whatever gene needs to be turned on or whatever gene needs to be turned off. It's going to be a message from the actual blueprint. And so we call it transcribed because we're just rewriting it. We're rewriting the messages found in the blueprint, and we're writing it over to the RNA, and we're using the same language. We're using nucleotides. Now, the next step is the RNA message gets translated into a protein. Translation, the assumption there is that we are changing languages, and in this case we are. We're changing from nucleotides to amino acids, because proteins, as we'll see in the next part of this lecture, proteins are made up of amino acids. And so what this slide's telling us is that the DNA encodes information for the amino acid sequence of proteins. Absolutely. The DNA is giving the information to the RNA. The RNA then is giving the information to the ribosome for the proteins to be produced. Now, DNA is what we call a double helix. So think about it as a ladder that's twisted. And so it's twisted into a helix. And so there's going to be actually two of these linear structures right here. So here's one side, and there'd be another side over here that we'll see on the next slide. So the base repairing rules tell us that A's or adenines bind with thymines and cytosines bind with guanines. This is important. If we go back and we look at what an adenine and a thymine is, adenine is a purine, thymine is a pyrimidine. So A's bind with T's, a single ring with a double ring. Cytosine binds with guanine, another double ring with a single ring. Now, we're going to see why that's the only way that they can actually bond here on the next slide. So when we look, you have a double ring structure. You have a double helix structure here. One is going to be going in the 3' direction, this way. The other one's going to be going in the 5, the 3 down to this side. So they're going in anti-parallel, which means they're running in opposite directions. The base pairing, as you can see there, adenine binds a thymine. It can make 2 hydrogen bonds. Cytosine and guanine can make 3. So that's why we can't get an adenine binding with a cytosine. There'd be one extra hydrogen bond right here cytosine's trying to make that adenine wouldn't be able to fulfill. 1 or 2 of those along the entire sequence of DNA is not necessarily going to cause too many problems. However, you have a lot of those along the entire sequence of the DNA, you end up having integrity issues, and the DNA is going to be unstable, and eventually, will be mutated. Mutations typically lead to terrible things like cancers. Cancers typically lead to death. So, having too many mutations is never a good thing. So, strong DNA is always going to have adenine and thymine bonded together, cytosine, guanine bonded together. So RNA, as I already mentioned, the most common of the RNAs are messenger RNA. We'll be seeing this more as we move forward into the other lectures. But for right now, we're just giving you the basics of it. So RNA is similar to DNA, except for it has ribose instead of deoxyribose. It has uracil instead of thymine, 2 things we've already pointed out. The uracil, the reasons why it's used in RNA, there's many reasons why. However, that's mainly saved for a biochemistry class that would be like a third or fourth year undergraduate course. Nothing I'm going to ask you any questions about in here. Just know that uracil is replacing thiamine in RNA. The biggest thing, though, is that RNA is a single polynucleotide structure. So it's not a double helix like we see in DNA. That makes sense because we're only getting the message off of one part of the DNA. I don't need both sides to get the information I need. I just need one side. So RNA is going to be single-stranded. So here's a little bit of a slide that shows you the differences between DNA and RNA. The same information that we talked about before, but a good spot just to make sure you have all your P's and Q's together in terms of the differences and similarities between the two. So other nucleotides that are available. We're going to see this a lot. Nature tends to be very conservative, and so in this case, instead of building a whole new molecule, what it's done is it's taken a molecule it already has and it's modified it slightly in the form of calling it adenosine triphosphate. So what this is it's adenine. Right here would be the phosphate. Here's its sugar. Here's its base out here. What's the difference is we have two extra phosphates attached here, which is where it gets its name, triphosphate. It's going to have three. ATP is considered to be the main currency for energy inside the cell. All cells use ATP. We won't call it adenosine triphosphate too many times. We'll just refer to it as ATP. The other two that we'll talk about as we get closer to chapter 7 and 8 is going to be nicotinamide adenine dinucleotide and flavin adenine dinucleotide. Again, two molecules that use the adenine nucleotide but have modified it slightly, in this case to become an electron carrier. And so we'll be talking a lot about electron carriers in the following lectures as we move forward. The third macromolecule that we're going to talk about are proteins. And traditionally when we think about a protein, we think about like meats or nuts or some other food group that has high protein, eggs. Here, what we're actually referring to is the biological definition of a protein, which I already referred to as being part of the central dogma, where the DNA is transcribed into RNA is translated into proteins. And so proteins can fit in multiple categories. In this case, this is not an all-inclusive list. This is just a list of the major categories that we tend to see proteins associated with. So one, of course, is enzymes. Almost all enzymes are proteins. We'll see that there are very, very few that are RNA in nature, and we'll discuss possible reasons for that as we get to that slide. But here, the easiest thing to think about is that enzymes are all proteins, 99.9% of them. Defense, this would be part of your immune system. Different molecules that are involved in helping fight off infectious diseases are going to be protein-based. Transport, in this case, hemoglobin is used as an example. Hemoglobin is found inside the blood. This is what carries oxygen and carbon dioxide around throughout the circulatory system. Support, this could be structures in your hair or your nails, such as keratin, that helps give support. We'll also talk about some protein fibers that are found inside the cell itself that are part of the cytoskeleton. Motion, this is what we typically think about when we think about meat. You're usually buying muscle tissue as, chicken breast or chicken thigh, chicken legs, hamburger steaks. This is all muscle tissue that you're buying. The two proteins that muscle is made out of is called actin and myosin. Regulation, almost all hormones are protein-based. And storage, this would be involving certain molecules in our body that store some of these harder to find elements, such as calcium and iron. Nowadays, we fortify, especially milk, with calcium and iron, but evolving, our body did not have access to either one of these in large quantities, so it found a way to essentially store or hoard calcium and iron whenever it finds it. So proteins all have the same basic shape. They all have amino acids as their monomer. And so there are roughly about 20 amino acids. It says 22 here. There are a couple that are modified, but there's 20 base amino acids. And these are going to be the basic monomer for the proteins. And so what we already seen when we were doing functional groups, we have the amino group right here. We have the center carbon. We have the carboxyl group over here. We have a hydrogen. And then we have this R group. R group stands for the restricted group. This means it cannot be whatever we want it to be. It has to be one of the 20 shapes that we see amino acids having. So in this case, glycine, it's very basic. It just has another hydrogen right there. And that would be the simplest of all the amino acids, all the way up to tryptophan, which has ring structures and is a little bit more complicated. But the same backbone will be found in all amino acids, the amino group, the center carbon, carboxyls, and this hydrogen. The only thing that will change is what's attached right here. So how they come together? Same way, dehydration synthesis. Essentially, we're going to have water removed. So we take the water out of the OH here in the carboxyl, combine it to the hydrogen here of the amine group, and it becomes water. Well, in this case, what's left is here in red, and this is the important part. This is what we call a peptide bond. And so this is going to be what is created between each amino acid as we start building these proteins out. And remember, these proteins can be hundreds, if not thousands, of amino acids all combined together. What's interesting about the peptide bonds is they're only found between amino acids, sort of like phosphodiester bonds are only found between nucleotides of DNA or RNA. And what's also going to happen is it doesn't allow any rotation right here. So the double bonded oxygen can't be up on this side because the hydrogen would be impeding it. So it has to keep this shape. That's kind of unique. So then we can talk about actually looking at the protein. If I was holding a protein in my hand, I can look at it in different ways. And so if I'm looking at just the sequence of amino acids, which amino acids are those made up of that make that protein? I'm talking about the primary structure. I'm just talking about which amino acids come in which spot along the protein. The secondary structures are going to be these different groups that we find on a regular basis in proteins. Two names that you see right there, beta sheets and alpha helixes, are the more common ones, but again, not an all-inclusive list. There's quite a few different secondary structures out there. In of themselves, the secondary structures don't tell us a whole lot. But if we move up and we start looking at them in larger context, then sometimes we'll start seeing that structure and function that we keep talking about in biology. The structure equals the function. And so if we have something of similar structure, and maybe it's a protein we've never seen before, but it starts looking a whole lot like one that we do know, then more than likely the function is going to be pretty much the same. Or at least it makes it for a good starting point for experimental design. The tertiary structure is going to be its final folded shape. And so this is going to be, what does that protein look like when it's all folded up and it's active? Now, the next one's quaternary. Quaternary means that we're talking about more than one protein working together. We call these molecular machines. And so we'll see a lot of different molecular machines as we go forward in the different chapters where there's more than one protein involved in making something happen inside the body. And so if we're referring to all the different proteins that are working in conjunction with each other, we call it the quaternary structure. If we're just talking about a single protein, we can talk about it in primary, secondary, or tertiary. So here, just another way to see the same thing. Now, when we look at those secondary structures, we can start, as I said, if they start coming together, we start seeing more than one of them all together. Then we start getting structure that we can start relating to other structures that we know of. So some ways to think about it are in motifs. These are going to be 2 or 3 secondary structures together. So what you see there is a beta, alpha beta motif, also known as Rossman's fold, helix turn helix, beta barrels. All these come together, and these, when we see these motifs in a protein, especially an unknown protein, as I pointed out, it gives us opportunities to try to understand that protein better and to give us some direction for experimental design. Domains are going to be functional units within large structures. So looking at the proteins, we're going to talk about how in an enzyme, there's really only one spot that's important, and that's what we call the active spot or the active site. The rest of the protein is mostly structure. support and whatnot. And so here when we're looking at domains, this is looking for the functional areas that are important for that protein to actually do its job. So the way I like to think about it is a car. Cars have domains and motifs and secondary structures too. For instance, if we were to look at the engine, that would be a domain of the car. The entire engine would be a domain. The different parts of the engine that come together to make the engine would be motifs. And then those individual parts that all come together to build the engine complete are going to be the secondary structures. That all comes together to make a domain. There are multiple domains in the car. You have the transmission also. You have the cabin where everybody sits. All these would be different domains that are made up of different motifs that are then even broken down even further, and we can look at the individual secondary structures. Think of a protein the same way. A protein is just one big, large structure that's made up of smaller structures called domains. Those domains can be broken down even further into motifs, and the motifs can be broken down even further into secondary structures. So here we can see that same thing. So here's a beta, alpha, beta. So we can see the alpha sheets right here. The beta sheets right here. You can see the alpha helix right here on the left, followed by some more beta sheets. So that would be a beta, alpha, beta, or that Rossmann fold. We also have the helix turn helix, where we have the alpha helix. We got a series of amino acids right here that aren't making any shape whatsoever. It allows for it to turn and go back on itself into another alpha helix. Those would be different motifs because they're made up of different secondary structures. Over here on domains, now look, we have one domain right here, we have another domain right here, and we have another domain right here. And looking at the motifs and secondary structures that put those together helps us understand how that protein may actually work. Some examples of a protein is a chaperone protein. As we talked about, protein folding is incredibly important to get to the proper tertiary shape. And so one of the things that we can look at is chaperones. Chaperones are special helper protein. You think about a chaperone, what's the definition? Chaperone is essentially somebody that helps assist something. And so here, chaperone proteins help the protein fold into its correct shape. So they're little helpers. They're helping those brand new foreign proteins, because when they come out of the ribosome after they've been translated from the RNA, the protein is going to be in its single primary structure, just a bunch of amino acids connected together. It has to be folded. Part of that happens in what's called the Golgi apparatus that we'll talk about next week, but also can also be folded properly by the chaperone proteins too. Some examples of how important these chaperone proteins are is the disease cystic fibrosis. Cystic fibrosis is a single genetic mutation. So in this case, just one nucleotide is off in all 3.4 billion of an individual that leads to this very fatal disease. And so what occurs in these folks is that they have chaperone proteins that are not able to fold the cilia correctly that are important for helping pull the mucus up out of the lungs. And so while they're making everything, it's not being folded correctly, and that folding failure leads to the manifestation of cystic fibrosis. So here's just an example of how it happens, very basic, but you can see here the misfolded protein going into the chaperone protein. A little bit of magic of ATP. Remember, that's the energy needed to make these reactions work. It's going to essentially fold it, and out it goes. Nice thing is, enzymes are not burned up, used up. They just keep doing their job. So once that protein is folded back correctly, it's ready to go back to the beginning and find another one and fold it up. So enzymes just keep going until the job is done or they've been shut off. They don't burn up or get used up. What happens when a protein falls apart? We call that denaturization. Denaturization means that we're removing it from its natural shape. And so a couple things can lead to this. pH changes. So if it's a protein that needs to be in an acidic environment, it finds itself in a neutral or basic environment, it will denature, or it could be vice versa. Being a basic amino acid being found in an acidic environment can also cause it to denature. Temperature is a big one. Heating something up too high can essentially cause it to denature. And different ionic solutions, so different salt concentrations can cause proteins to denature. All right, the last of the four macromolecules we're going to talk about is going to be hydrophobic lipids. The term hydrophobic means that they do not mix well with water. So hydro being water, phobic means to have a fear of. So here, fear of water. They're not scared of water, but it's a term that we give them when they don't like to mix, otherwise known as being insoluble. Insoluble means that it doesn't mix well or doesn't dissolve. Why is it that lipids are so hydrophobic? Well, it has a big part of being the nonpolar. So remember, nonpolar means that there's no charge. If it's polar, it means it's positive or negatively charged. Nonpolar carbon-hydrogen bonds. All those carbon-hydrogen bonds, all the way back to the second slide of the lecture talking about hydrocarbons. All those carbon-hydrogen bonds, there's nothing for it to bond to. All the valence electrons have been found a partner in the form of hydrogen, and therefore this molecule has no interest in working with anything else. It's just happy on its own, therefore it becomes hydrophobic. So the most common inside biological organisms is triglyceride, the tri meaning three. What is it that it's three of? It's three of the fatty acids. So fatty acids are the monomer for lipids. And so they are not identical. Their chain lengths vary. So chain length is going to be how many carbons are connected to it. Some of them are 18, some of them are 20, some of them can be different numbers. So the three fatty acid chains on a triglyceride do not have to equal each other. But they do have this glycerol, which is this 3-carbon molecule right here with the oxygens. That's a glycerol, and it is what's connecting the fatty acid chains. Now, some other terminology for lipids are going to be saturated versus unsaturated. When we use the word saturated, that means every spot is filled up. And so in this case, a saturated fat has no double bonds. That means every single structure right here. So look at all these carbons. Granted, part of the glycerol has this double bonded oxygen, so ignore this one. But looking over here, all these carbon and hydrogens are all single bonded. That would be a saturated fat. That tells us it's probably animal origin. Unsaturated fats have one or more double bonds in them. So imagine that maybe here between these two carbons, that's a double bond, which means that we'd lose these two hydrogens. Now it's unsaturated because we double bonded here, and that's going to lower the melting point, so it takes less energy to break those bonds. And we typically see that in plant origins. So people that tell you, oh, unsaturated fats are healthier for you, or getting your fats from plant origin, one of the reasons why is that melting point. Animal fats take more energy for our body to break and to utilize versus unsaturated fats that have these lower melting points, therefore less energy is needed to break them up. Trans fats are produced industrially. There's an entire story that goes along with trans fats, but the biggest point to trans fats is that they are now deemed illegal. FDA finally banned them a couple years ago in the United States because of the fact that they are not very healthy. They are industrially made, and our body doesn't know how to deal with them. So many folks that would eat trans fats would find themselves sick, allergic reactions, other adverse reactions. A lot of people, they could eat them, no problem. But ultimately, there's enough people that were having issues that they ultimately banned them in our foods. Now, the most important besides triglycerides is phospholipid. Phospholipid gets its name from having a hydrophilic head, and so we'll draw them in different ways. In this case, we draw the phospholipid right here with the hydrophilic head, and then two fatty acid tails. So these are going to just be fatty acid tails right here that give it a hydrophobic property. So when we think about a phospholipid, it's almost like a bipolar molecule. Part of it's hydrophilic, the other part of it's hydrophobic. Well, this is important for how cell walls form. We're going to talk about cells in chapter 4, but then we're going to have an entire chapter dedicated to membranes in chapter 5, as well as in lecture. We'll have an entire lecture dedicated to membranes. So it's important for you to get these phospholipids down now. So the easiest way to draw them, as I'll draw them up on the board, is right here on your left. Here in the middle is going to be the way to draw out the molecule if you want to know what elements are involved. And over here on the right is just a ball and stick model. So if you're sitting there looking at this going, what am I looking at here? Well, you're looking at the same exact structure, just drawn 3 different ways. Same thing here. We got it drawn 4 different ways. But here's the most important way to draw it. This is what we look at as a cell membrane. So notice what's happening here. The cell membrane is going to be the membrane that goes around the outside of the cell, also known as the phospholipid bilayer. So bi just meaning 2. And so see, notice what happens here is that the tails face each other because they're hydrophobic. The inside of a cell is going to be made of cytoplasm, which is mainly water. The outside of the cell is going to be also a lot of water. Our body's mainly water. And so when phospholipids find themselves in aqueous solutions, the tails naturally face each other. The heads are going to face out towards the water. And this ends up being a really good barrier for molecules to be able to not be able to penetrate to get inside the cell. The cell then becomes selectively permeable. It means that it's able to select what it allows it to bring in and out of the actual cell, which we'll talk about more as we move forward. So in summary, carbon's a framework. You have 4 covalent bonds that can be made based off of 4 valence electrons in carbon. You have the different isomers, stereo and structural. You have chiral, meaning basically mirror image. You got the carbohydrates with that very specific carbon double hydrogen back to regular oxygen 1:1 ratio. They come in different forms, but mainly sugars and starches. The monosaccharides are going to be the monomer. The starch or sugar, or actually the starch is going to be the full structure, in terms of the polymer. But they can do both energy sources and structural support depending on which one we're talking about. Proteins, lots of different categories. You got the primary, secondary, tertiary structures. Quaternaries when we're talking about more than one protein working together. Chaperones as an example of a protein that helps fold other proteins. Nucleic acids, DNA and RNA, made up of the 4 nucleotides, guanine, thymine, adenine, cytosine. In RNA, the uracil replaces thymine. Phosphodiester bonds are going to be the important bond that's formed between nucleotides to form the nucleic acids. ATP is a nucleic acid. Well, it's not a nucleic acid. That's wrong. It's a nucleotide. It's adenine that has formed with 2 extra phosphates on it. And lipids being hydrophobic, you got fatty acids as the monomer. Triglycerides is one of the important ones found inside biological organisms. But most importantly is the phospholipid bilayer, which is going to be the membrane that forms around not only the cells, but a lot of the organelles that we find inside our cells. All right, well, I hope you enjoyed this, and we'll see you next time in class.